we've been able to use one of our very cool new toys, which is a time-lapse microscope. And so I've been making some movies of that process in action. And hopefully this will play, and I can show them to you now. So what you see here, I'll use the mouse to point, this is a mixture of neutrophils. These are normal neutrophils, and they all contain vitamin C. And they've just been left in a dish, and they're going to undergo that cell death process. And I'll point them out as they start to ruffle and bleb. You'll see them. This short video covers a period of about 5 hours. So, you know, it's time lapse. So we see it here. Here goes the first one, there. I think this one's next, there. There's another one down here. There he goes. See him go? There's another one here. I think that one, or this one up here is going to go next. That one's gone. There he goes. So one at a time. There's one just coming into frame. There he is. So you can see they're normally moving around. They undergo the cell death, and then they stop moving. So, and they do it. You know, they're on a program towards doing this. And those cells at the end of that process, or during that process, they're supposed to be sending these signals to the macrophage. Now, from these same preparations, we've also got a preparation of macrophages. And so what I've done is I've taken those dying neutrophils and I've added them to this plate here. And these cells that you see here, these are macrophages. They're all stuck on the bottom. I want you to concentrate on this guy here. He's amazing. So I'm going to show you what happens when we put these dying neutrophils onto this cell, and remembering that diagram, what you're going to see is you're going to see the cell eating the cells in the vicinity. And so there he goes, you watch him go. So you can see inside the cell, you can actually see the shape of the cells it's taking up. So they're mostly kind of ghostly, the dying cells, but he's hunting and you know, looking for, you know, you'll see him pick up, you know, cells from this vicinity here. So, and then he goes off and hunts up in this direction for a while. And you can see other cells around doing very similar things. So this process starts up as soon as we add those dying neutrophils to that mixture. They're amazing. I think I sat there for about 4 hours looking at it. I don't know how many cells they can eat, but it looks like an awful lot. And so inside that, they're recycling all of that material that they're taking up. And this is what's happening in your bodies pretty much on a daily basis. So we wonder what happens if you have a vitamin C deficient neutrophil. So if the cells don't have vitamin C, how does this affect that cell death process? And so these are vitamin C deficient cells, and they're at the same age as those first ones you saw that are all that bubble and boil, so to speak. And so what you see here, if you watch this one here, these cells don't do that. There's an awful lot of activity inside that cell, but there's no blebbing or bubbling, and in fact they pop. So, and at the end of our time, so you watch this one here, it's kind of halfway through this process, they start, instead of shrinking up, they start to swell up, and then they form, you get these kind of balloon cells. And at the end of the process, we've got a whole lot of these cells that look dead. But so what happens to them if you put them with the macrophages? What would happen to them? And so here we have another lot of macrophages, and these are, so here we are up close. And so if you concentrate in this area here, there's two candidates that look very similar to our previous one. And you'll see that we've added, so I've added those dying vitamin C deficient neutrophils to these. And you can see that these cells, they're definitely alive, they're sitting there, but there's no activity with respect to recognising those dead and dying vitamin C deficient neutrophils. So these cells are blind to that signal. And what that means for people is that we need good vitamin C when we're sick. We need good vitamin C for our immune cells in order for that cleanup to happen in a controlled way. And as we were thinking about this, and here's a summary of this part of the talk, we were thinking, so vitamin C is required for effective neutrophil removal and cleanup. And actually, what would happen if this went wrong? And so we thought, do we have any clinical evidence that that would be devastating? Because someone asked me a question in one of our research groups once, and they said, what would happen? And I said, you would dissolve from the inside. And then we thought, what happens to people with scurvy? And actually, you know, here's an example of someone with scurvy. Your tissues break down from the inside. So, you know, we think it's very possible that one of the major contributors to this kind of damage is the fact that your neutrophils are bursting and releasing those enzymes into your tissues in a way that's actually devastating. And as a result of that, you know, people with scurvy die sudden and horrible deaths. So it was in thinking about this and this aspect of our work that we thought, well, this is true in neutrophils which have vitamin C, but how's the vitamin C doing this? What's the mechanism behind this? And we started looking at other kinds of neutrophil functions, and one of the papers that came out around the time that we were thinking about this showed us that a lack of oxygen, so if neutrophils are active in a lack of oxygen, they have plenty of vitamin C, they behave almost identically to our neutrophils that have no vitamin C, even though ours have got plenty of oxygen. And so we started thinking, you know, what's the link here? Because what the cells are doing is exactly the same thing. And are these two things linked in some way? Sorry, I need to go back to that. Because this is Usha Pujari, who's a PhD student. She's now studying the effect of hypoxia or low oxygen on neutrophil function, turning out some very nice data too. Now the link, taking you back then to we look at proteins inside cells, the link is a protein called HIF1, hypoxia-inducible factor, or HIF. And this protein controls the cell's response to low oxygen under normal conditions. So every cell in your body has it, and it's a stress response protein that will react in the absence of oxygen. So, and it does that in this way. So the protein's made up of two components, alpha and beta, and the alpha component gets modified by these enzymes called prolyl hydroxylases and others, a few others. And these enzymes stick these hydroxyl groups on that protein, and that means it gets destroyed. So this is an off switch. And these enzymes need oxygen and vitamin C. So this is the link. This is the link between vitamin C and the white blood cell functions. So if oxygen or vitamin C is missing, then what you have is this protein is stabilized, so it stays in the cell, and it is able to pair up with its sister protein, and that upregulates a whole lot of gene responses that means that cells can overcome the stress of low oxygen.